Thanks so much, John, and thanks also to ALK Australia, ALK Positive Australia, TOGA. It's really sort of great to be able to share this work and have this work sort of really generously supported by the donors as well. So I'm really quite grateful for that. As John mentioned, so it's important to acknowledge the traditional owners of the land which I'm broadcasting from. That's the Wurundjeri people of the Kulin Nation, and pay my respects to their elders past, present and emerging. Now a little bit about me. So I am a biomedical scientist and a bioinformatician by training. My PhD was done at the University of Melbourne back in 2018, and since then I've worked at the Peter MacCallum Cancer Centre here in Melbourne, in Professor Mark Dawson's laboratory, where broadly my work is interested in understanding how genes are regulated in cells and how that goes wrong in cancer. And the work that I'll present today is focused on lung, of course, but we also do work in blood and brain as well. And so the lab as a whole is led by Mark Dawson and his wife, Sarah Jane Dawson. We're based in Parkville, in Melbourne at the state-of-the-art VCCC Comprehensive Cancer Centre. Our lab is a relatively large group. It's a very multidisciplinary group that consists of not only scientists, but clinicians and computational biologists and several students as well, ushering in that sort of next generation. And broadly, sort of as is shown on the slide, these are the sort of areas that we focus on studying. Now, the overall goal of my work is really to address this question, to try and overcome resistance in cancer, drug resistance in cancer. And really how we do this is by an integrated approach that combines, innovation in technology, which can increase the number of questions and the type of questions we can ask. And then that really feeds into two really fundamental discovery layers. One is, as I said, understanding how genes are regulated, and that is at a very fine detail. And then looking more from, I suppose, a top-down layer at how populations of cancer cells interact with one another and respond to treatments, and how they might differ in their treatments, is sort of the other aspect. We kind of come at this from both a bottom-up and a top-down approach, and the overall goal is obviously to discover targets that we can develop into novel therapeutics that can not only enhance and be combined with current treatments, but provide enhanced benefit for the future. So today I'm going to talk a lot about clones, and not these type of clones, for any Star Wars, aficionados in the audience, but specifically these type of clones. So when I say clone in this presentation, I'm referring to a population of cells, a population of cancer cells that have all originated from one single cell. And so they are, in effect, clones of each other. They're genetically identical. And, they share some behaviors that I'll describe in this talk. And the reason for this, and we sort of focus on looking at cancer through a clonal lens, is because we know cancer is a clonal disease. It arises from a cell that is normal. That cell, whether by chance or exposure to various, mutagens and so on, can acquire mutations in its genome, and that can lead to a malignant transformation event. Now, what happens is, as the cancer grows, more mutations accumulate, and this causes different sort of subpopulations or subclones, if you will, to exist within the same tumor mass. So if you look at it at a macro scale, it looks like a tumor, but within that there's different populations, and we call this subclonal genetic diversification. Now the sort of classic pathway is, upon diagnosis, patient will receive a treatment, they'll undergo a therapeutic response, and ideally what will happen is we'll enter this phase of minimum residual disease, where there is sort of the maximal level of therapeutic response has been reached, and hopefully the cancer is almost undetectable at this point. But of course, what we know happens, and this is not just lung cancer, it's essentially all cancers, is that various things can happen at that point that means that not every single cancer cell is eliminated, and that gives the cancer an opportunity to change and adapt and eventually relapse. And so what we know now is that it's this population of different clones that can give rise to some different properties. Some might cause a metastasis. Some might drive relapse at the primary site of the tumor. And so a fundamental question for us is, what are the actual features of these different clones that explain why they behave in different ways? Now, there's two main models to try and understand this that we know in the field. One is a genetic model, where, as I talked about, mutations accumulate in DNA, and this can lead to resistance to therapies. A drug that hits a particular factor that's mutated in cancer, if that factor requires a mutation that stops that drug from binding, of course, that's going to lead to resistance. And so this is a genetic model of tumor evolution, if you will. Where largely we focus is actually what's become more well appreciated recently, which is non-genetic evolution, which is similar adaptation, but it happens without changes to DNA. And it happens because of changes in the pattern of gene expression, which genes are turned on or off, and to which level in different cancer cells. Now, what we're sort of finding is that these are not mutually exclusive phenomena, right? These are interdependent, and actually different cancers and different sort of individual subclones within a person's cancer might sit somewhere differently on this spectrum, sitting maybe more towards a genetic model or more towards a non-genetic model. So there's a real push in the field to try and understand how these different properties can be explained by either genetics or the non-genetic models. And one way to do this is to use a tool called cellular barcoding. Now, this is essentially a set of tools that can label individual cells. So you can actually put a different tag into each different cell, and that tag can be inherited by the progeny of that cell over time, right? And so we can construct something and we essentially call this process barcoding the cells. Now we can use other tools, such as inert viruses, to introduce those barcodes into the DNA of cancer cells, so that when that cancer cell divides, that specific barcode that it has been given is inherited by its daughter cells. And essentially, the result is something that looks a bit like this, where if you imagine different clones now, demarcated by the different colors, you can see how the primary tumor is made up of a few different clones. This purple one here, because we can trace these barcodes over time, we can show that the metastasis is primarily this purple one. And this metastasis down here is driven largely by the green one. And so that's really the power of barcoding, that we can trace them over time. Now, the simplest experiment you can do is really to barcode some cells. And now cells that have a different color are essentially a single clone that has a single barcode. And when you apply a treatment, you can sort of see which cells become resistant to that. So in this case, it's the red one predominantly and a little bit of the blue one. And as I said, we can then count up all those barcodes and figure out which clone was the one that became resistant. So the power of this is that we actually get clonal identity, because that's inherited through cell division, and we can read that out later. We can understand the fate of each clone, what happened to it after we put the treatment on, because that's also traced over time. But what would be really nice to have is actually an appreciation of the pattern of gene expression, which genes are on, which genes are off, and in which clones, both before and after treatment, to really understand why this red clone is the one that sort of developed resistance. Now, we have developed techniques for this as well. So the main one that we use in our lab is called single-cell RNA sequencing. And this is sort of different from the previous technology, which was to extract all the RNA, which tells you inside a cell which genes are being expressed. The more RNA of a particular gene there is in a cell, the higher that gene is being expressed. Previously, we would just take the cells, mash them all up, in a tube, and sequence the RNA. Okay, and that would give us expression value for every gene, but we wouldn't know, if there was 10 different subclones in there, for example, how much of gene X is being expressed across those different clones. So the innovation, about 10 years ago was to be able to now sequence individual cells. So now you get an expression value for every single cell. And the real beauty of that is, in this sort of Lego diagram, you now move from, having a mashup of Lego, and you don't know how many of each piece there are. You don't know sort of really like what color and what type of piece you've got. Now you've effectively got information on every single piece. You can count up each one. You can figure out exactly what makes each one tick. And so that's the real power of single cell sequencing. But the problem with single cell sequencing is that you actually don't understand from it very easily. You can do this but it's not very easy to do. You don't understand these lineage relationships that I talked about, which cell gave rise to which other cells, how they're related in a clonal sense. On the other side, I've just told you that the barcoding methods, the cellular barcoding methods, give you this information. So through the barcoding, we can read out how cells are related to one another. But of course, as I've just said, we don't know the pattern of gene expression in each clone, in each cell. So really what we set out to do a few years ago now is to develop a method that can combine both of these pieces of information. And that tool that we developed, we published this in Nature in 2022, is a tool that we call Splinter. It stands for Single Cell Profiling and Lineage Tracing. Lineage tracing is in effect a different term, an analogous term to cellular barcoding. Now, how this works is pretty simple. We have a construct that is inserted into the DNA of cells, contains a random barcode. So it's a random sequence, and that is a unique barcode that each cell is going to get a different one. Now, the data can be read out in two different ways. One is, as I said in the example before, is just counting up all those barcodes and seeing, okay, we've got 20% of the population is clone A, 10% is clone B, and so on. That's the genomic DNA approach. The real utility of this and the real innovation is that the barcode is converted into the RNA, okay? And so what can happen is that when we do single cell sequencing on a cell, on a cancer cell, we can figure out the pattern of gene expression in every single cancer cell, and we can also detect what barcode that cell has from the RNA. So we not only now get the lineage information, we also get the gene expression profile, or the cell state, as we call it. And the beauty of Splinter is that it now marries these two pieces of information. So why is this powerful? Well, it actually allows us to understand how a tumor is evolving, across a pattern of treatment. So if we barcode cells with Splinter, we want to understand, and then say we give those cells a treatment, and it's this purple clone in this one, in this particular example, that has become resistant. We want to know, because it's actually really important from a treatment perspective, we want to know if the propensity for that purple clone to develop resistance was already there before we put the treatment on. That's pretty important to know if you're going to give a certain therapy. Is the cancer already predisposed to being resistant to it? or is that resistance acquired as a result of the therapy? So Splinter allows us to understand exactly this by using single cell sequencing of a pre-treatment time point. We call this a baseline. And in a situation where the sort of response you're looking for is pre-existing, what you'll find is that this is a representation of how single cell RNA sequencing data looks. Each little dot here is a different cell. And you can essentially interpret this by saying that cells that are closer together have a more similar gene expression pattern. Okay, so this gray cell at the top here is very different in the terms of genes that are on and off in that cell to these red cells down here. But more importantly, what you can see is that if all these red cells belong to a clone that became resistant, you can see that even before the treatment, they had a very different gene expression profile to all the other gray cells, the cells that were actually killed off by the treatment. And that's really important to note. The second example is the opposite, where it wasn't pre-existing in the population, but it was actually acquired as a result of therapy. And you can see here now these red cells, the ones that develop resistance, are actually no different from all the other cells before the treatment is applied. So it's an adaptive response of the treatment. So we use Splinter really across a whole range of questions, from understanding drug tolerance to understanding how frank resistance develops, and also in terms of trying to understand the subtle differences in how cancer cells behave and how that might impact different outcomes, such as treatment, such as, which clone is going to drive a metastasis, and so on. So today I'm going to talk about some sort of work that we have in the pipeline at the moment that's really focused on understanding drug tolerance. And it really, boils down to this question, right? How does differences in how clones behave impact the ability of a cell to tolerate a different therapy? Now, of course, we're working on non-small cell lung cancer. In particular, we're working on the ALK positive subtype. And it's no should be familiar to the audience, I'm guessing, sort of how this cancer comes about. Now, ALK, or the anaplastic lymphoma kinase, as its full name is called, is actually a cell surface receptor normally. So it has a domain that sits outside the cell, sort of projecting outside the cell, and it's going to normally respond to signals that come from outside the cell and tell that cell, okay, I need to do a certain thing. The signaling from ALK normally will feed into growth-related pathways. It will largely tell that cell, I need to divide and I need to grow. Now, what happens in ALK positive cancer, ALK positive lung cancer, is that the most common alteration is what's known as an EML4 ALK fusion. Now, this occurs on chromosome 2, which is sort of shown here. Now, the EML4 gene is sort of sitting this way on the chromosome. ALK is sitting this way. And what happens in this cancer is that there's an inversion in the chromosome. This is a specific type of mutation that can happen. And that brings part of EML4, basically chops both of these genes in half and brings part of EML4 and fuses it onto the ALK gene, and the opposite happens as well. And so the oncogenic, or the cancer driving sort of variant, is called this EML4 ALK variant, and that accounts for 85% of ALK positive non-small cell lung cancers. And the effect on ALK is that it's now no longer sort of sitting outside the cell. It's now fully inside the cell, and it's just signaling constantly. It's what we call constitutively active. And there's essentially unrestrained, or the brakes have completely been taken off the signaling through this, and it drives a very potent growth response. Now, we've developed multiple generations of tyrosine kinase inhibitors against ALK. In particular, one that we work with is lorlatinib. So this is a third generation inhibitor. It's marketed as Lobrina in the US. This is a sort of what ALK looks like from a protein sort of wireframe standpoint. And you can see how this molecule just sits inside the active site of ALK and essentially stops it from performing its normal signaling function. Now, the clinical results of lorlatinib have been very, very impressive. Not only can this compound rescue, this is in a preclinical mouse model, I believe, rescue resistance to first-generation compounds, such as crizotinib, but the recent trial results, have shown, big increases in sort of key metrics such as progression-free survival up to the 5-year mark. And so this is now FDA approved in the advanced setting, with more trials being undertaken using this as a frontline therapy. Now, there's a key problem, and it's sort of almost the elephant in the room when you're talking about results such as that, is that essentially killing 99% of cancer cells isn't good enough, right? We need, it's essentially this situation where you have a weed growing in your garden, right? And you want to get rid of it. But you need to get rid of the roots as well as the stem. If you leave something behind, then it gives the opportunity for relapse. It gives the opportunity for resistance. And so we have been very interested in understanding these, roots, if you will, that remain after a very potent therapy such as lorlatinib. And these are called drug-tolerant persister cells. And in effect, they have very adaptable properties. They're able to change and withstand the treatment in various ways. And it's essentially trying to overcome these cells is the main hurdle to a more durable therapy. Now, this is sort of a schematic of how these drug-tolerant cells come about. As I've explained earlier in the talk, after a cancer initiates and grows, it undergoes that clonal diversification. The therapy comes on, and then we have a response. The tumor shrinks, and then there's a tolerant population that remains. This is a population of tumor cells that is still there, still present, but is not being killed by the drug, is sort of remaining static, ready to relapse, ready to grow back out once a sufficient signal or condition is met, right? A condition for resistance. It could be a new mutation is acquired that stops lorlatinib from working, It could be other signals that change how the genes are expressed in that cell. So for this work, what was really key is that we try and understand these questions. How is the drug tolerance, as I mentioned, pre-existing or acquired? What do the drug tolerant cells look like? And do they have specific properties that we can target to specifically kill them, as well as the rest of the cancer that we can already target with something like lorlatinib? Now, the way that we model this is pretty simple. We've sort of focused on an ALK positive cell line. So this is a human cell line. We call it H3122. So this has a EML4 ALK fusion of the variant 1 subtype. And we've conducted a lot of experiments in conjunction with a more common mutation found in non-small cell lung cancer, the mutations in KRAS, specifically the KRAS G12C subtype. And so essentially what we do is we model all of this disease in mice. So we will barcode the cells, we will put them into mice and establish a tumor, and then we will treat those mice with either the respective agent. So lorlatinib for the ALK variants, and in the RAS setting, there's also RAS G12C inhibitors available, such as sotorasib. And the real idea here is to try and understand pre-existing versus acquired modes of resistance, as I mentioned. The first thing to note is probably one of the most striking things I've ever seen, in that if you look at the clones that form tumors in these mice, and you do that across multiple independent mice, so you're looking at it, at least I'm showing 4 examples here, but we've done this, 16 different times. almost every single time what you see is that the clones that cause a tumor are exactly the same in every single animal. And you can see that as shown on this sort of bubble plot, as we call it, where each dot is a different clone, so a different population of genetically identical cells. And the size of these individual bubbles reflects its contribution, its relative contribution to the tumor size. There's also a correlation matrix here, which in effect shows you that the clonal makeup of these tumors in these different mice is approaching 1, right? Very, very similar. The interesting thing came when we now looked at the single cell RNA sequencing data. And so for the ALK positive line, what we found is that, similar to my example from the start of the talk, the dots that are in red are the cells, or the clones, I should say, that cause the tumor, okay? And you can see that they're very interspersed with the ones that are in blue, right? And the ones that are in blue are the ones that did not cause the tumor, okay? In contrast, you can see in this KRAS model that the ones in red, the ones that are forming the tumor, are completely distinct from the ones in blue, the ones that did not cause the tumor. So in one model, this is a pre-existing mode of, this is not resistance because we haven't applied a treatment yet. This is tumor formation. These cells are primed to cause aggressive growth inside a mouse. Whereas in this ALK situation, it's adapted. It's not something that already exists when we grow the cells in a dish. Now, we could show that that wasn't due to the genetic makeup of the cells, because when we sequenced the entire genome of these ALK positive cells from the tumors that arose in the mice and then compared them back to the original population, essentially what we found were no genetic changes, right? So there was no new mutations that happened in those cells that allowed them, that gave them an enhanced ability to form those tumors. The opposite was true in the KRAS line. We found many mutations. So we have this very interesting dichotomy where in ALK EML4, ALK positive situation, you have almost 100% non-genetic adaptation to tumorigenesis. Whereas in the KRAS setting, it's almost completely predetermined by the genotype or the genome, the different mutations that those different clones have. And some clones just happen to have the right combination of mutations that allow them to drive aggressive tumor formation. So now if we layer in treatment on top of this, it starts to get a bit more interesting. So we essentially developed a system where we could induce the formation of drug-tolerant persisters by essentially repeating the same experiment I've just described. We establish the tumors in mice, and then we give one group of those mice a control treatment, or we call it in the lab, we call it a vehicle, which is just corresponds to the solvent that the drug is dissolved in. Or we gave lorlatinib. And so what we found, if I just skip forward, is that in the black group here, these graphs represent the size of the tumor as it grows in the mouse. The black group is the control group, the one that hasn't been treated. You can see these tumors just continue to grow, and they reach a very large size by the end of the experiment. But in the lorlatinib treatment group, essentially from the start point of treatment, we can see that those tumor sizes rapidly collapsing, and they sort of collapse and then they settle into a static sort of situation here that's around 100 sort of also 150 cubic millimeters. So this is small enough to still be sort of palpable, but as you can see from the graphs there's no real change over time in these tumors. They're just sort of flatlining. They're not growing and they're not dying out. So these are drug-tolerant tumors. Now, when we did the same barcoding experiment on these tumors, what we found was also mind-blowing. Because what we would have predicted is that actually this treatment would have killed off a whole bunch of clones. Okay, we should have, the tumors have shrunk so much that multiple of these clones we would have predicted would be completely eliminated, and there should be a residual population of, a subset of those clones that were able to survive and form a drug-tolerant tumor. But essentially what we found was the complete opposite. Almost every single clone, you can see in these bubble plots, again, these are this vehicle is the control. These are the clones that formed those tumors in the untreated setting. In this drug-tolerant tumors, you can see it's almost exactly the same repertoire of clones that are present at almost exactly the same size as well. So what's happening? And I want to shout out to John for this example, for this sort of analogy, because this, I think, sums it up perfectly. It's like one of these toys that maybe you're familiar with, where the tumor starts off, it's treated, and bits of it don't fall away. The whole thing shrinks, and it all shrinks coordinately and in the same, in a coordinated way, where each clone that makes up that tumor is now still present at the same size, right? And that's just something that we find is completely mind-blowing, how this is happening in such a coordinated way. Now, what's really interesting is to try and understand the gene expression profile of those drug-tolerant tumors. Now, you can see, again, if we extend out this experiment and do something that's called a drug holiday, where we actually take the therapy off and we ask, well, can the tumor regrow? We find that in every mouse, the tumor rapidly grows back out in this drug holiday situation. And you can see from the single cell data that's shown in the middle that the cells from drug tolerant tumors in red, the cells from the untreated tumors or the vehicle treated tumors in black, and these drug holiday tumors are completely separated. So in terms of the genes that are being switched on or off in these drug tolerant cells, it's almost completely different to the untreated setting. It's not, completely unexpected, because the drug does induce different changes like that. But what is sort of surprising is that if you look at these, the ones in blue, the drug holiday, they've seen the drug, and now we've taken it off, and they completely revert back to what they were like before they saw the drug. So again, this is reinforcing this adaptive mode of resistance or drug tolerance. And that's sort of shown here, where they adapt in this way, and then you take the drug off and they fall back to sort of very similar to an untreated tumor. And as I said, there's a range of gene expression patterns that are associated with that process, and most of them are related to how the cells are sort of conserving or creating or utilizing energy. Because one of the main pathways that ALK is signaling down is an energy generation pathway. So if we kind of kill that, the cell has to find other salvage methods to be able to provide enough energy for it to survive. And so these are some pathways that we're investigating further at the moment. Now, the second thing we looked at here is actually to try and understand this drug tolerant population in a bit more detail. Okay. Now, if you kind of look at this sort of, again, single cell RNA sequencing data from the ALK positive line, you'll notice that there's these little projections in the data. Okay. So these purple ones here and here and here, these are all sort of shooting off in different directions. They're suggestive that there's actually some gene expression differences even within the drug tolerant tumors themselves. Now, what we know of the lung and the different cell types that make up the lung is that there's actually quite a lot of cell types. And, the cell types in the sort of main windpipe, the trachea and the bronchi, are sort of slightly different to as you get further down into the smaller bronchioles, and then in the alveoli are made up of a sort of different set of cell types called alveolar type 1 and 2. Now, we wanted to know, we actually got interested in this because we noticed when we looked at I should mention these different normal lung cell types that I'm showing have a completely different gene expression pattern. And that basically tells the cell, I'm a AT2 cell, I'm a ciliated cell, I'm a neuroendocrine cell. And that's a very strong identity. But what we actually found in these drug tolerant cells is that they were sort of spreading out and adopting features. They are not any of these cells. The cancer cells are not any of these normal lung cell types, but they're adopting features of the normal lung cell types. And we think this sort of diversification process is a key mechanism that the drug tolerant cells are using to withstand and tolerate the treatment. Now, again, the beauty of Splinter allows us to look at this from a clonal level, okay? And I showed you earlier in the talk that in the drug-tolerant tumors, they have exactly the same clones, and those clones are at exactly the same size as the untreated tumors, yeah. But we have this bit of a paradox. We have very similar clonality, but we have clear differences in the gene expression profile of these different cells that comprise this drug-tolerant tumor. But when we layered in the barcoding information on top of this, what we could actually see is that, for example, we saw different clones. So again, this is groups of cells that share a common ancestor. Different clones were biased to respond to the drug in different ways. So for example, this clone here, 98089. Okay, the number doesn't really matter too much. That's just the barcode that it happened to get. It is, you can see most of the cells that belong to this clone in green are actually sitting in this ciliate-like region of the drug-tolerant tumor. So that clone is biased to adapt to the drug by adopting features of a ciliate cell. Now, we found examples of clones that behave in similar ways, but for different cell types, such as the basal AT1, AT2, the neuroendocrine subtype. And actually, this is one we're very interested in because one mode of resistance to these inhibitors that is thought to be non-genetic is what's called a transdifferentiation to a small cell lung cancer subtype. And that is largely thought to be driven through a neuroendocrine pathway. So the cells must adopt features of a neuroendocrine cell in order to undergo that resistance mechanism. So we're very interested in cells that are adapting in this way. And of course, there were features of other cell types as well. So the key takeaway here, it's sort of pretty complicated data, but the key takeaway is from our models, the drug-tolerant tumors are not a single, uniform entity. They're actually a mosaic. They're a mosaic of these different adaptive states, these different ways that the cell can adapt by taking on these different lung cell type features. And that seems to be directed by the clonal identity, or it seems to be clonally directed. So essentially, cells that share a common ancestor will adapt in the same way. So if we're thinking about trying to eliminate this population, we need to find ways to target all of these states, right? It's not just going to be enough to be able to target DTP cells that are ciliate biased, for example. So finally, in the last 5 minutes or so, this brings me, so the work that I've just described, we've been working on for a few years now. Really the work I'm going to talk about now is the work that's been generously funded by ALK Positive Australia and TOGA. And that's now to extend on this finding by trying to identify the so-called Achilles heel of these drug-tolerant cells. Now, there's a few ways that the field has tried to go about this. The first is to try and identify cells that are going to become a drug-tolerant cell before this sort of idea of a pre-existing gene expression profile. Our data would suggest, given that essentially every clone can adapt, that this is maybe not, at least in the models that we've looked at, maybe not the best approach. So we are taking a different approach. we are going to force the cells through treatment with lorlatinib. We're going to force them into a drug tolerant state. We know through the experiments I've just told you about how they're going to adapt, the different possibilities of how they can adapt. And then we're going to ask, what factors do those cells require to keep them alive? I discussed how they need to, they need to come up with a new way to provide energy for the cell, for it to survive, for example. There's a few different things that the cell will need in order to survive in a drug tolerant state when its primary method of energy and growth signals has been cut off by the treatment, by lorlatinib. Now, the way we're going to do this is to use a technology that's called CRISPR. So I think probably it's famous enough now because it's completely revolutionized molecular biology and biomedicine that a few people in the audience will know about this, but this is an ancient bacterial defense system against invading viruses that we have been able to harness, the scientific community has harnessed for a whole range of applications. And that's because it can specifically and in a programmable way edit the genome, okay? So it relies on this protein called Cas9. There's various types of these Cas enzymes, but the most common one is Cas9 that along with, in yellow, what's called a guide RNA, will bind to a specific place on the DNA and cut it, okay? And what that allows us to do is essentially cut out entire genes. So if we want to know the function of, gene X in disease Y, we can use CRISPR, we can go in and cut out gene X in cells that have disease Y, and we can see what happens, okay? Now, the proposal that has been funded by ALK Positive Australia is to do this in a very systematic way. So we're essentially going to take genes one by one. The human genome contains about 20,000 genes. We're going to essentially take these genes one by one, knock them out one by one, and then we're going to ask, does knockout, for example, of gene A cause the drug tolerant cell to die? Right? Can that potentially be, a synthetic target that we could harness to try and develop a new therapy against these drug tolerant cells? That is in effect what we're trying to do here. And in pilot screens that we've done, so we've sort of got some preliminary data here. The way to read this data is essentially what we call a hockey stick plot, where things above the line, these are genes that when they're lost, they actually promote resistance to lorlatinib. So we obviously don't want those. We want things that fall below the line, things that are depleted and that actually in the drug tolerant cells promote sensitivity. Yeah. And so what we found in this interesting work is that an example of a gene called KEAP1 that behaves differently in a different context. So if we model the resistance in the lab by just growing the cells in a flask and then treating them with lorlatinib, if you knock out KEAP1 in that context, if you get rid of KEAP1, the cells are actually resistant to lorlatinib. And this is actually a known resistance pathway because many people have done a similar type of experiment. So this was actually kind of a control arm. Really what we wanted to do is something that is more relevant to the actual disease biology. We wanted to model this in the mice as I've been doing with the experiments I've talked about today. And actually when you do that in a mouse and you lose KEAP1, it actually promotes sensitivity to lorlatinib. So these drug tolerant cells that lack KEAP1 are more sensitive, will be preferentially killed off by lorlatinib. And so what we think is that there's now sort of a bit of a theme in the field that things that are modeled, in the lab in the way that I'm describing on the left-hand side do not necessarily always translate to how biology works in vivo, as we call it, in the body. So we think that, for example, KEAP1 might be an in vivo specific dependency in ALK positive lung cancer. And there is some clinical data to back this up. If you look in the TCGA, the compendium of all the cancer patients that have been sequenced, and you look in two very important clinical trials that looked at different treatments for ALK positive lung cancer, you almost never find a KEAP1 mutation in a patient that also has an ALK that is also ALK positive, suggesting that there's a reason why these things don't occur, because actually KEAP1 loss or KEAP1 mutation is actually detrimental to the survival of that ALK positive lung cancer. So to try and summarize, I think I'm coming up on time. Really, what we've tried to show with this early work, and we're now going to extend upon with the proposal, is to now try and understand the principles of how drug tolerance not only emerges, but is maintained using ALK positive cancer as an example. So what's really clear from this work is that the tolerance is actually acquired after you put the treatment on. If you take the treatment off, it's fully reversible. The cancer will start to grow again. But also what I've shown is that the drug tolerant cells are not uniform. They're not all the same. They are what we call a heterogeneous mixture, okay, of cells that will respond to the drug in different ways. And I've shown you how, with the power of our splinter tool, we can show that that is directed by the clonal origin or the lineage of that particular cancer cell. And now we're using these in vivo CRISPR screens that I mentioned to try and extend upon and find some of these dependencies that we can use to specifically target this drug tolerant population. So I'm pretty excited to get started with this work. I think it's going to be an exciting time now that we have essentially all the tools ready to go, that we can pull the trigger on this and hopefully find some really cool, not just cool biology, of course, but targets that can potentially be translated in the future into real world patient benefit. So I'd like to just thank a few people, of course, ALK Australia and TOGA, and the members of my lab. So the work I described has been co-led by another postdoc in the lab, Suniat, and of course led by Mark. John's been instrumental in the last sort of 6 months, particularly for the proposal and shaping that. And then there's also a team at Pfizer who have enabled us to do a lot of these studies with the lorlatinib compound, and various support staff at Peter Mac, without which we could not have done the sort of cutting edge work that I've described today. So I think that's all from me. I'd love to take some questions if there are any. Thanks.
I'll let you take a moment, Dane, and maybe have a drink of water while I just quickly say, every time I see you present, you just continue to blow me away by the progression in the knowledge and understanding that continues to develop in the work that you do. Like every time you seem to move another step forward in the understanding, and I feel incredibly privileged that we've been able to come into your work at this point in time with such a body of knowledge. Like, you've already done so much work on that splinter, identifying the barcodes, and I mean, you've already started to dip your toes into how to understand and knock out the genes and all of that sort of stuff. I feel like we've come in at the glory end of a lot of hard work where answers are possible, and likely, rather than one of those projects where people go, I've got a great idea and I don't know if it's going to work, and I'm going to try a whole bunch of things and we'll see what happens. I feel really hopeful that you're going to come up with some responses. I guess one of my first questions is, how many of those 20,000 genes do you think you can actually get through? It's a lot of genes to look at and try and knock out. Is there a way that you can get a shortlist, prioritize a certain bunch of those genes so you don't have to look at 20,000 and get to answers quicker?
Yeah. Yeah, that's and thanks, John. I'll just say that I'll echo what you said. I think at the start, the chance meeting that we had, sort of really kicked off this work, because we sort of had the pilot screen data I presented at the end was almost just sitting there, and we didn't really know what to do with it. And then that through those meetings, we really sort of galvanized this new project. So I'm really excited about it. The screening approach, is complex when you think about trying to do all 20,000 genes. Okay, yeah, totally. Like it's scaling that and doing it inside a mouse. Remember, like it's actually feasible to do in a dish if you're growing the cells, because you can just grow millions and millions of cells. But the way we do it, particularly in vivo, is, as you said, by shortlisting the genes. And the way we do that is essentially by a theme. We know we have a pretty good handle on the function of most of the genes in the genome, or at least the most important ones that we think are involved in, a lot of diseases and this sort of thing. So the first subset of genes that we did in that pilot screen was actually any gene that has been shown to be targeted by a drug that is approved by the FDA. Okay, because we wanted to go straight for the jugular. We wanted to see, is there a gene that when you lose it, you affect the survival of the drug tolerant cells, and there's already a drug you can just go bang and hit it with, okay, and see if that provides a benefit. Now, we might have found something from that, and we're definitely following up on a lot of, there's the Keep One story, but there's also other findings from that screen that I think are really interesting. The second sort of, cab off the rank in terms of a set of genes will be things that are involved in that adaptability that I talked about. This ability of the drug tolerant cells to turn on a different program when they're exposed to the drug and then turn it off and switch back on their normal sort of growth program. And that's sort of a broad group of factors called chromatin factors and transcription factors. And these just happen to be the type of factors that we work on most in the lab. Okay, these are proteins inside the cell that affect how the DNA is read. They affect how the DNA is interpreted and turned into a response by the cell. So you can imagine that it's going to be those proteins that are really important in regulating the adaptation to the therapy. And so we're hoping that's a pretty broad class of proteins. So the first short list is about 2,000. So we're going to tackle about 10%. But, there's, I think, a pretty good chance that we'll find something quite interesting, even in that smaller subset. And the fact that it's smaller means that it's much more feasible to do in a shorter time frame.
So if I understood you correctly, are you saying that first group that you're talking about, you're going to try and test genes that there are already known FDA approved medications out there for, and if they're effective, there is the potential to fundamentally repurpose that medication for use that may knock out that gene and halt resistance in ALK?
That's essentially the idea. I mean, there's sort of many steps, as you want in that, between this hopefully discovering that association and then hopefully being able to repurpose a treatment in that way. And we were not really discriminatory here. Essentially, anything that has been FDA approved, we added to this list. The other thing to keep in mind is that some of these therapies are sort of targeting multiple things at once. So there's kind of a bit of complexity there that I glossed over, but essentially that was the idea of the first screen, as you've said, is to try and find what we call a rational combination strategy that might have benefit.
A great way to shortcut to treatment, but I won't hog. I'll give the opportunity if anyone else has any questions. Feel free to unmute yourself and ask a question. If you can, as I've seen John just do or whatever, turn on your camera so Dane can see who's asking the question. But is there anyone who wants to put up their hand and ask a question of Dane while we've still got some time? I see Lisa's put up a hand. Good use of the hand function, which is down the bottom of the screen, a little hand. You can click on that if you want. Lisa, please go ahead.
Yeah, thanks, John. And thanks, Dane. That was an amazing presentation. I'm so excited to see what the outcomes are of it. I was really kind of blown away as well by those patterns that you showed, and also the fact that when you looked at different types of lung tumours, like the KRAS versus the ALK, the similarities there. I just wonder, and also that interesting picture of the different parts of the lung and how it sort of dominates and tries to evolve according to that. I'm just kind of wondering, though, if there's been any consideration of looking at metastatic sites like the brain, which is a really, crucial area for ALK positive lung cancer patients, and if there's any similarities, differences, and the rationale behind that kind of, treatment pathway that becomes, what's dominant in that process that it causes it to go to the brain.
It's a fascinating question. There's a lot of research in a range of cancers to try and understand why, in particular cancers, some cancers are prone to metastases to different bodily sites, right? And that's kind of an active area of investigation. Some people have shown that it's related to the metabolism of the cell. We sort of, are not directly looking at the question that you've asked. I didn't really discuss this, but when you're trying to model these diseases, there's inevitably pros and cons to every type of model. And a way that we chose to model the disease, which is one where we could really perform all of that exquisite sort of barcoding that I showed, is to model it essentially as disease that has moved outside the lung cavity. Okay, so sort of we're modeling it as extrathoracic, disease. Now, it's not quintessentially metastatic, although if you actually look in the gene expression patterns of the cells that form tumors, they have a very what's called an epithelial mesenchymal sort of signature, which is something that is upregulated in metastatic cells. So we do think to some degree we are looking at aggressive extrathoracic disease. We haven't actually looked, and fundamentally the cells are still lung cells. Like I sort of started to show that they adopt these different features of the different normal lung cell types, but the fundamental identity of those cancer cells are of a lung cell. So I think still if they were to, end up in the brain or somewhere else, they would probably still have, they would maintain that identity, but they would take on flavors. I don't know if it went to the brain, if, they would take on flavors of, say, different brain cell types, but I think they would modify, the various sort of compendium of lung cell type sort of gene expression profiles that they have available to sort of suit that environment. Yeah. And that's really what we're interested in. So we're kind of taking a site agnostic approach, if that makes sense.
Yeah.
We're trying to firstly just catalog all the different ways in which they can adapt, and then use that information to try and pair it with the screens to try and hit all of those different possibilities.
And I suppose then the other thing to consider is you're looking at variant 1 and you have variant 2 and variant 3. So are they the same? Are they different? There's so many factors, but you've got to start somewhere. And I just want to say thank you for the work you're doing. I'm really excited to see, how it all pans out.
Great. Thank you, Lisa. Appreciate that.
I see you've got your camera on, John Cannings. Is that because you had a question you'd like to ask?
It wasn't so much a question. Can you hear me, John?
We can.
It wasn't so much a question. I had a little bit of difficulty getting my microphone back on. I wanted to say thank you, Dane. Great presentation. You covered a lot of area there, and you are right. Drug resistance is a major issue for ALK positive patients and most non-small cell lung cancer patients on TKIs. I suppose the question, if I have one, is about the gene expression, whether the protein expression is in the cell or on the surface of the cell. Does that make it any different for the type of research that you're undertaking to identify these resistance cells?
Yeah, thanks for the question, John. So you're talking about the ALK protein, aren't you? And how I mentioned that in its normal state, it's sort of part of it is sitting outside the cell. So the most common way ALK is rearranged is, as I said, with the EML4 gene as part of that inversion. Now, what happens is the product of that, none of it sits outside the cell anymore. It's all internal, okay? And that affects its regulation because it's now decoupled. Like normally it would receive a signal from outside the cell, and that would tell it to actually send a message inside the cell to activate those growth pathways. But through the mutation, it's now completely decoupled from that. That extracellular part is no longer produced properly, and it's no longer linked to the rest of the ALK protein, which actually is the business end, which does the messaging. So the product is that sort of mutated protein just keeps signaling, it's just active all the time. Now, Lisa mentioned the different variants, and there's also different partners that ALK can fuse to. It's not always EML4, but it's mostly EML4. Sometimes, I don't know if it's fully appreciated if it matters which is the fusion partner, just that decoupling event happens, and you now have sort of unrestrained activity of this kinase. Where it will come into play with the research, I think, is in understanding, as Lisa mentioned, the effect of the different variants, because the mutations can be slightly different parts of ALK can be retained or lost, by those different variants. And that might have important functional properties. We've just looked at variant 1, so I can't comment on what those would be at the moment, but they have been shown to sort of act slightly differently in response to treatment and so on. So I think the plan for us would be, once we kind of hopefully find some very promising targets, we would then be able to test the different ALK variants, okay? And we would be able to then understand how, in sort of hopefully more detail, how the different variants might not only respond to the primary therapy, such as the lorlatinib, but respond to maybe a combination that has, a new therapy that also tries to come in and target that drug tolerant subset. And I would be surprised if we didn't find any differences. That's just the complexity of biology. In an ideal world, we would find something that hits all variants, equivalently with equivalent potency. That's always the goal for anyone who's doing this sort of work, but it's not always the case. So hopefully that answers your question. And yeah, thanks very much.
Look, in the interest of time, I think we'll leave it there. We've already gone over a little bit. But I have to say, Dane, like your research, really hits at the heart of, I think, what's really important to most people with ALK is, if we can find a way to manage resistance and make the TKIs that we have access to at the moment effective and durable in the long term, that is a huge step towards, at least making ALK a chronic disease that can be managed in the long term rather than what it is at the moment. And I can't wait to have you back in a year or two's time and to hear the results and find the exciting answers that you might find. And hopefully there might be a target or two that looks really favourable, that's already got a medication that's been approved that might short track us to finding a combination to stop resistance and helping us stick on our TKIs for really long periods of time. I think everyone is as grateful as I am for the work that you do and that you've chosen to make ALK a focus of your research, because it is the research that gives us hope. So thank you very much for taking the time to share that with us. And we look forward to hearing from you further, and good luck with your research. I hope it goes well.
Excellent. Thank you so much, John. Thanks everyone who tuned in. And yeah, I very much look forward to sharing some of the results of this work. In the hopefully not too distant future. Yeah, definitely.
Good luck, mate. Thank you very much.
Thanks very much, everyone. Bye-bye.